Hello there, you're welcome back to Biology Video Lectures with Akerile Olademeji Philip. It's my wonderful pleasure to have you for the lecture today. So we'll move on the series on organization. We've done a number of topics already, so we'll just move on to this. Now we're moving on to enzymes and metabolism. It's a section, all right? So we're looking at how enzymes help in metabolism. Basically, enzymes, we know they are biological catalysts. Metabolism means the summation of all the activities, the breaking down and the building up of substances in our body. So let's get started. So basically speaking, it says here that digestive enzymes work outside the cell. So most enzymes are secreted outside the cells to bring about their effect, so to say. Now, what did they do? They help to digest the large insoluble molecules into smaller ones, which can be absorbed in the bloodstream. Then what's metabolism? Metabolism, on the other hand, is sum of all reactions happening in a cell or organisms in which molecules are synthesized or broken down. So when they are synthesized, we call that anabolism. And when it is broken down, it is called catabolism. So the summation of anabolism and catabolism forms the word metabolism. So it's just saying you're building things up from the scratch or you're breaking them down. These two summed up forms metabolism. So we said that enzymes are what? Biological catalyst. Catalyst from chemistry means it speeds up the rate of chemical reaction. So when we say biological catalyst, it means it's a chemical made within the biological system, within the living cell, then it speeds up chemical reaction. So it says here it allows reactions to occur much faster than they would have without the enzyme. So in fact, it is said that without enzymes, it can take days for the food we have eaten to digest. Yes, that's really, really true, because when people age, the rate of digestion reduces. Yes, so that's because the volume of enzymes in old people is lower, so it takes a longer time for the digestion to happen. But digestion still happens anyways. So I'm trying to say that part to make you understand the fact that without enzymes, it's going to take a very long period of time for our food to get digested, so to say. Now, substrate. Now, the thing I want you to understand here is that what is being digested is called substrate, and the enzyme remains the enzyme as well. So substrate temporarily binds to the active site of an enzyme. So we're going to look at that really in big time in this lecture here. So this is an enzyme, for example. Then the food, the substances to be digested is called the substrate. So that will occupy the space. So that space is the active site, so to say. So it binds to it temporarily, then afterwards it disengages. By the time it's going to disengage, it's going to disengage in a broken form. It has been dissolved already or worked upon. Now the good thing is that the enzyme remains the same, both at the beginning and at the end of the reaction. It doesn't change. The same shape, the same size still remains the same, so to say. And enzymes can work with a number of substrates at the same time. Some can work with as much as 100, as much as 1,000 substrates per second. So that's the reason why, like I said, our food gets digested within hours. But without that, it's going to take days for food to get digested. So this is what we're trying to say here. So you see that this is the active site, this is the substrate. So this substrate will bind, you can see that, into the active site, this shape like this. You can see it's actually fit into it. Afterwards, the reaction occurs, then what happens? Enzyme is freed, it remains the same, the shape is the same, but the product has been released. So you can see here it was just a whole sum up of structure, but now it has broken down into this and into this. So that explains how enzymes remains unchanged from the beginning of the reaction and at the end of the reaction, and how it breaks down substrates into simpler ones. So enzymes are very, very important in humans, in all organisms, basically. So here, I'm trying to show you something here that enzymes are specific. Remember to pause the video every now and then to write things down. They are specific in their action. What does that mean? It means that an enzyme will not allow another substrate which is not meant for it to fit in. For example, here, substrate 1 looks like, I don't know, like a T. This is the enzyme for substrate 1. Excuse me. You agree with me that it is easy for this to fit into the space here. But substrate 2 cannot fit in there. Why? Because the shape will not. So it means that every enzyme has a particular substrate it is meant for, or you can say every substrate is meant for a particular enzyme, either way. So this explains the fact that there is something called specificity of enzyme and its substrate. Yes. So it says here, the enzyme 1 can bind to its substrate 1 as they are complementary to each other in shape, so they can easily fit in. But substrate 2, sorry for that spelling mistake on that, substrate 2 is not. It can't fit into it. So that's how enzymes work. So every enzyme has the substrate to work for. Now you can check this out also. You can see the active site allows a particular substrate to fit into it. If the shape is altered, I mean, if the shape of the enzyme is altered, yes. For example, because in the course of this lecture, I'm going to show you that there are factors that can actually alter the shape of this enzyme. Then, much more than that, even if the shape of the active site of the enzyme remains the same, you must make sure the right substrate is fitting into it, so to say. So here, let me read some of these things to us. It says the specificity of an enzyme is a result of complementary nature between the shape of the active site and of its substrate or substrates, so to say. So enzymes are proteinous in nature. They contain amino acids joined together to form protein, like I said, like that. All right. Now, if the order of the amino acid determines the shape of the enzyme, so if the order is altered, the resulting dimensional shape changes. That's what I was trying to tell you, that there are factors that can change the shape of the active sites, like temperature, like pH, a number of things really, acidity, like I said. So, but I want you to please pay attention here. You might want to pause the video here and write things. A lot of information here will be found useful for use, all right? So